Yeah, good morning and thank you very much, Professor Atre, for starting this award series today. Let me introduce Professor Anirban Banerjee. Anirban is currently an associate professor in the Department of Biosciences, Bioengineering. And from what I see from his CV, he has really come a long way. So he started out by getting the Young Faculty Award at IIT Bombay in 2012. Then, he made his mark in teaching, where he got the Excellence in Teaching Award from IIT Bombay in 2017. And in the same year, he also got the Early Research Achiever Award from IIT Bombay. So you can see that it has not been a one-day journey for Anirban. It has taken several years, before the research has built up to this level. That today he's, in 2021, he's getting the Research Publication Award from IIT Bombay. Professor Banerjee's research has led to path-breaking advances in the field and has established new paradigms in infectious diseases. And this is by challenging existing dogmas. These endeavors have not only led to novel discoveries, but have also invoked critical new questions needing sustained research efforts to solve. And this is crucial because, if you keep doing that, you will keep getting bigger. The magnitude and impact of his research is evident by international and national press coverages such as Science News and recognitions as the best published scientific paper in the life sciences from India by the well-recognized Cell Press. So that's actually a very, very big achievement in TNQ technology. And this has also led to Professor Banerjee securing several extramural excellent research funding. So once again, let me congratulate Professor Anirban Banerjee for this huge honor and excellent award. And I'm sure that he'll go on to do very, very good work in the Department of Biosciences, Bioengineering to be recognized by very, very big agencies in India. Thank you very much.
Thank you, Rohit. Anirban, please upload your slides, please. So you got a 5 research publication award, and the next one with Professor Devavrat Maiti. I just request him to be ready, followed by Professor Velega, Professor Anurag Garg, and Professor Sandeep Saha in this slot. Yeah, Anirban.
So thank you, Rohit, for this kind introduction. I appreciate. Thank you to the jury members for selecting our work as one of the awardees for this, and congratulations to the rest of the awardees in this year. The title of our work is a little creepy, but hopefully you will be able to appreciate that by the end of this talk. So the title is Why Make Enemies If We Can Be Friends? And I'm primarily going to be talking about this bacteria, which is shown over here, which is known as Streptococci pneumoniae, which is primarily known to cause pneumonia in children as well as adult population prior to SARS-CoV-2. Before I start my presentation, I would like to acknowledge all the members of my lab, my fellow colleagues in the department, without whom it not be possible, and these 3 individuals, Manali Surve, Anjali Anil, and Smita Bhutra, the 3 stupendous PhD students because of whom this award is possible. So it belongs to them and not to me. So I'll start something which is typically known in the infectious disease biology is an arms race. That means a bacteria which is typically known for us to cause disease tries to trump us by secreting several different factors. They can be some toxins, they can be some other soluble mediators such as hydrogen peroxide, and there can be some other proteins which the bacteria also secretes to compete killing other microbes residing in the similar area. While it is doing so, the host is also not sitting idle. The host actually calls a lot of macrophages, neutrophils, the professional phagocytes into that particular space to kill off the bacteria. That is what is typically known as inflammation. The host also secretes a lot of antimicrobial peptides, a lot of antibodies, and all these to trump the bacteria. So there is this constant war going on between the bacteria as well as the host, and each one of them trying to trump the other. And while that happens, there is a huge amount of inflammation and the damage that occurs in that particular tissue space. And beyond, after the infection has been cleared, there will be a repair phase as well as the resolution phase, which is essential for the homeostasis of this tissue. That means the tissue goes back to its normal level. Now, under some conditions, this repair phase or the resolution does not happen in a proper regulated fashion, and that results into sustained level of inflammation at that particular tissue, which is typically known as dysregulated inflammation, and courtesy to COVID, which you all know right now. And that's what actually leads to majority of the cases a lot of mortality, because this dysregulated inflammation inflicts a lot of self-damage on the host cell. So that brings us to this particular question, is that what is a good bacteria and what is a bad bacteria? So the good versus bad, and as you might know that our body, there are several different and several numbers, plethora of bacteria that is present. In fact, in our body, there are more number of bacterial cells than our own cells. So now if at all you look at this graph, if there are pathogen load, sometimes the bacterial load is tremendously high. And because of that also, the host does not feel sick. The health of the host is really good. And that is typically known as the good bacteria. And in some cases, even though the pathogen load is equal to that of the good bacteria, but the person feels sick. And that's where, which is known as that, this is called as tolerance of this good bacteria. So we are not mounting immune response against the so-called good bacteria, but we are mounting immune response against the so-called bad bacteria. Now, this becomes particularly important in the case of the bacteria if they have only the human host as a reservoir, because if the host is sick or dead, then that would lead to loss of reservoir for the pathogen. Now, that brings us to choice, that is Streptococci pneumoniae, and it's an obligate symbiont. By obligate symbiont, I mean to say it has primarily only one environmental reservoir, which is the human nasopharynx, and that's where it stays as a commensal bacteria, does not cause any harm. But time to time, it goes to the lower respiratory tract and actually causes pneumonia over there. Now, generally, from this human nasopharynx, there it forms in the biofilm-like state, as you can see over here in this particular picture. And from there, what happens is that time to time, there is a dispersion of the biofilms, and it transmits from one individual to another individual through these air droplets like this. It has been known in the field for quite some time. And so this is the only route for transmission from one host to another host, and understandably, the bacteria, it wants to spread to as many number of hosts as possible because it wants to increase its number of reservoirs. So now, if there is no other transmission possible through other body fluids, such as the blood, such as the fecal oral route, that essentially means that the causing an invasive disease where the host would actually succumb or die, that actually is a dead end for the pathogen, or for this particular pathogen. Now, for this transmission to happen from one host to another, there needs to be a light bit of inflammation. And that inflammation is induced by a toxin molecule secreted by the bacteria, which is known as pneumolysine. We will refer this to as PLY henceforth. Now, this inflammation is critical because it leads to transmission to new host, but also leads to a new paradox, is that if there is dysregulated inflammation, which earlier I have talked, that leads to mortality and loss of occupiable habitat. So how this inflammation is regulated among the different strains. And you can see among the different strains of Streptococci pneumoniae that are present in the world, there will be different forms. And there are 20 different forms of this particular toxin which is present across the different Streptococci pneumoniae present in the world. So that brings us to this question, if this particular protein toxin is so important, why there are so many different variants present across the world? We will talk about only two alleles, allele 1 and allele 5. Allele 5 is present in the primarily in one particular form of the bacteria that is globally more present, globally more prevalent, and that generally does not cause lethal infections. So it only causes coughs and sneeze, but it does not cause pneumonia-like features where the host actually succumbs. And there are only 3 amino acid substitutions and 2 amino acid deletions out of 471 amino acid. And because of these substitutions and deletions, this particular allele 5, which is typically known as PLY-NH, and that's how we are going to refer to from now on. NH stands for non-hemolytic, H stands for hemolytic. So Where allele 1 will be PLYH, allele 5 will be PLYNH. So PLYNH is completely devoid of its pore forming activity. It does not cause the lysis of the host cells, and that's why it does not cause probably inflammation. So we first did an animal experiment, and as expected, the PLYH containing bacteria, which is given by these black lines, all the mice actually succumbs to the infection, whereas this green line shows that the PLYNH containing bacteria does not succumb to infection. But what is critical is that if you look at these PLYNH harboring mice at extended time point, 168 hours after the bacteria has been introduced into the mice, you will still see a lot of bacteria that is present in the lungs of these particular animals. That is given CFU per micromilligram of lungs over here. So now, courtesy to again COVID-19, you know that there is cytokine storm and all that thing. So in all cases, we see that the PLYNH containing bacteria have severely less cytokine expression or cytokine storm going on in the lungs compared to the PLYH containing bacteria. And the lung sections also shows that the air sacs are completely filled with the professional phagocytes in case of hemolytic PLY containing streptococci pneumoniae versus non-hemolytic PLY containing streptococci pneumoniae, where you see nice air sacs that's actually leading to gas exchange and the animals can survive. So if we look at the lungs of this particular bacteria, what came out to the fact is that PLYNH containing bacteria is predominantly present inside the lung cells. Now, streptococci pneumoniae is generally not to be going inside the host cell. It is a typically extracellular pathogen. So it is extremely surprising that this bacteria was able to find in a viable state inside the lung epithelial cells at prolonged period of infection. So that's what brings to our notice that what is that this PLYNH, the so-called non-hemolytic PLY, is conferring, so that the bacteria is now becoming predominantly intracellular in its life cycle or lifestyle. So indeed, you can see that the PLYNH containing bacteria actually is more invasive into the host cell, and if we see that the streptococci pneumonia can take different routes to invade the host cells, and we can see that one of the major host routes is this particular caveolin dependent pathway, and pneumolysine actually does this PLY disrupts this particular pathway, which is shown by these immunofluorescence micrographs over here. So in a nutshell, what I am trying to say is that whenever there is a bacteria which contains PLYH, it does not take up the lipid raft mediated endocytosis, but only takes other routes of endocytosis, because it actually damages the lipid raft containing endocytic pathway. Whereas non-hemolytic PLY containing bacteria can adopt both the pathways, and that actually leads to improved penetration inside the host cells. Now, getting inside the host cells is not sufficient, because intracellular defense mechanisms are there, particularly in the form of autophagy and other cellular mechanisms by which the host cell can tackle the bacterial infection. And over here, since PLYNH containing bacteria does not cause any sort of lysis on the endosome, you will be able to appreciate the fact that the PLYNH containing bacteria does not get killed by the predominantly autophagic process, which is the predominant process of killing intracellular bacteria inside the host cells. And that is also reflected by increasingly higher amount of the bacterial burden that is present inside the host cell with the bacteria which which harbors PLYNH compared to PLYH. So now, whatever I have shown up until now is that the PLYH containing bacteria goes inside the host cell in less numbers, and once it goes inside the host cells, it is typically killed by the host autophagic mechanisms, whereas PLYNH containing bacteria not only goes inside the host cells at higher numbers, it is also not getting killed by the host cell, just because of the fact it is not forming these pores on the endosomal membrane, so that it is getting protected. So now, with all this, while we are doing that, we were sure that how this PLYH containing bacteria that essentially cause disease also. So while we were doing this study, we also found out that the PLYH containing bacteria, there is extreme heterogeneity in the PLY expression among the isogenic bacterial population. As you can see from this immunofluorescence micrograph, there will be several bacteria which is not even expressing this PLY, while all are expressing this constitutively expressing protein, which is HLPA GFP, as you will be seeing over here, and so on and so forth. And what we found out is that is this low PLY expressing bacteria is primarily able to cause the disease in case of this PLYH containing bacteria. So PLYH containing bacteria has a subset of the population which is reflected by low PLY as well as extremely high PLY expression. And high PLY expressing bacteria is the one which is inducing inflammation, whereas the low PLY expressing bacteria is causing the infections. So from there, we hypothesized whether this low PLY causing or low PLY expressing bacteria is the ancestral population for this generation or evolution of this non-hemolytic PLY form that we have earlier discussed. And for that, what we did, we took a PLYH containing bacteria and subjected them to the in vitro in vivo evolution. That means we put them into the mice and actually allowed them to cause pneumonia, and then isolated the bacteria from the lungs of the mice and put them back onto the mice. So basically, we took out the transmission cost from the bacteria, and by doing that, we have generated 10 different lineages after 20 passages into each different lineage. And what we found out is that out of the 7 lineage, out of the 10 lineages, 7 lineages in case we see lower amount of PLY expression happening from the wild type, which is given over here, this black bar over there. And if we select 3 of the lineages, PLY, P21, P22, and P2010, and you will see in all these evolved strains, higher amount of the bacteria that is present over there in the lungs of the animals, though the animal does not succumb to infection, and their air sacs are much more better or free to do gas exchange compared to the PLYH containing bacteria, where the air sacs are completely filled with professional phagocytes. So essentially, coming to the conclusion, what we have shown is that the opportunistic pathogen Streptococci pneumoniae was having some mutations in the possibly the PLY regulators that actually results in the reduced PLY expression, which is typically representative of the persister population, which is our in vivo evolved clones. Further mutation may lead to non-hemolytic PLY-like phenotype, which is the PLY-NH, and further loss of virulence factors may lead to a commensalism at the different alternate host niche. Now, why Streptococci pneumoniae is going towards that particular direction is possibly because of the antibiotic and the vaccination. We are trying to take Streptococcus pneumoniae from its original home, which is the human nasopharynx. So it has 2 options: either it has to go extinct or it has to search for an alternate home. And this serotype 1 pneumococci, which is globally more prevalent, and of course one of the most prevalent serotypes in India also, this has these different pneumococcal pneumolysine variants, which shows mild disease and with low mortality, and that is primarily because it wants to preserve the host numbers. And it has no nasal colonization phase. It just comes, causes mild infections, causes coughs and sneeze, and that's how you suffer from mild infection, and then it goes to another host. And all these is possibly allowing Streptococci to acquire a new lifestyle supported by getting a new home, possibly in the human lungs. And over there, longer persistent it can do and actually lead to latent kind of infection, and finally it may lead to antibiotic tolerance and resistance like phenotype. So this adaptive evolution of Streptococcus pneumoniae from possibly it leading it from an extracellular pathogen to an intracellular pathogen, and that's what is our future studies in this direction. And finally, I would like to thank my group, all the group members over there, my collaborators, and the funding agencies, and thank you all for your kind attention. If at all you would like to have any questions, I will take that. Thank you.